Today's lab is cell division, meiosis, and sexual reproduction, which begins on page 81. Please look at the objectives on this page. So in this lab activity, we're going to look at meiosis and sexual reproduction in imaginary creatures called beboppers. Figure 1 shows a drawing of these creatures. In Table 1, the genotype of 6 different traits for each bebopper parent is listed. You are asked to fill in the phenotype for each trait. You can obtain that information from the area below the table. So let's do the first one together. The mom's genotype is A1A0. That's the genes that she has. What phenotype will she have associated with those genes? In other words, what will her physical characteristic be? If we look down at the bottom, we can see that the genotype A1A0 is associated with the phenotype of having one antenna. So we would write one antenna in this space. Please complete the rest of Table 1. So these two sheets have drawings on them that represent the chromosomes from either the mom, shown in pink on the left, or the dad, shown in blue on the right. Following the instructions on page 84, we're going to cut out all these chromosomes. So you can see that we've cut out all the mom's chromosomes. And what we're going to do now is take the chromatids that are identical and attach them using a paperclip. So we're looking for sister chromatids. So how do I know that chromatids are identical? Well, they're going to have the same length and they're going to have the exact same letters on them. So these two chromatids are identical. These are sisters. So I'm going to link them together using a paperclip. So the place where I'm linking them together, that's like the centromere. Okay? So there's now one duplicated chromosome. So I'm going to do that for all of these. Okay, so I've now attached all the sister chromatids using a paperclip. These are two sister chromatids, and these are two sister chromatids. They're not all 4 sisters together, just the 2 that are attached are the sisters. So how many chromosomes do you see right now? Well, remember when we count chromosomes, we count them by the centromeres, so there are 1, 2, 3, 4, 5, 6, 7, 8 chromosomes shown here. Page 84 now tells us that we need to pair up homologous chromosomes. Remember, homologous chromosomes are different than sister chromatids. Sister chromatids are identical and attached. Homologous chromosomes are not identical, but they are a matched pair. Homologous chromosomes are matched by size and by the genes they carry. Now, they don't carry the exact same forms or alleles of the gene, but they have the same genes in the same place. So these two chromosomes are one homologous pair. They're the same length, and we see they both carry the gene for L at the top and B on the bottom. Now, there's different forms of these genes. This one is the recessive lowercase allele. This is the dominant uppercase allele. But it's the same letter. So these two chromosomes are a homologous pair. These two chromosomes are a homologous pair. These two are a homologous pair, and these two are a homologous pair. Okay, so we've cut out the dad's chromosomes. Let's now pair up the sister chromatids, which remember are identical, exactly identical. So here's a pair of sister chromatids. We're going to join them together at the centromere with a paperclip. Okay, let's do that for the rest of these. Okay, so let's model meiosis in the mom bebopper. This is the process that's going to produce gametes, which, since this is the mom, will be eggs. So we have all of our chromosomes lined up by homologous pairs. So we've paired up our homologous chromosomes. So I hope you can see that. So how many chromosomes are in this cell? There are 8. Since this is meiosis, we expect that the daughter cells, which will be gametes, will have half the number of chromosomes as the parent cell. So the gametes should each contain 4 chromosomes. Okay, so what stage of meiosis is this cell in? We would say this is metaphase 1 because the homologous chromosomes are lined up at the metaphase plate. Now, it doesn't matter what order we put these chromosomes in as long as we pair up the homologous pairs. If you reverse these 2 chromosomes, that would be fine as well. Notice that there are several different ways we could arrange these chromosomes, and that possibility increases genetic diversity in the offspring. In other words, that means that every gamete this mother produces will not be genetically the same. Please pause the video now and draw these chromosomes, along with their alleles, in the cell in metaphase 1 on page 88 of your lab manual. So let's now go through what anaphase 1 would look like. What happens in anaphase 1? That's separation of the homologous pairs. So imagine our chromosomes are moving along the spindle, and now we've separated our homologous pairs. Now let's imagine telophase 1 occurs, followed by cytokinesis, and now we have our first cell division in meiosis. And let's put our chromosomes into those initial 2 daughter cells. These 4 chromosomes go here, and these 4 chromosomes go here. So this is metaphase 2. And it looks a lot like metaphase of mitosis because the centromere of each chromosome is lined up on the metaphase plate. Let's model what happens now in anaphase 2. What process occurs in anaphase 2? Separation of the sister chromatids. So there's anaphase 2. Now imagine telophase occurs with cytokinesis, and our chromosomes end up in different daughter cells again. Remember, we started out with 8 chromosomes in the parent cell, and after meiosis, we now have 4 chromosomes in each daughter cell, which is what it should be. Because remember, meiosis is going to produce daughter cells with half the chromosome number as the parent cell. In beboppers, the diploid number was 8, so we would expect 4 chromosomes in the daughter cells, the gametes, at the end of meiosis. So the haploid number is 4. Now let's take a look at these 4 different daughter cells. Let's take a look at these daughter cells. Here's gamete 1, gamete 2, gamete 3, and gamete 4. Are they identical? So we're going to randomly select one of these daughter cells to get fertilized. So there's our haploid egg cell. All right, let's go through meiosis in the dad bebopper to produce his gametes. So here are the dad's chromosomes. This is metaphase 1. The homologous pairs are aligned at the equator. Please note that the last pair of chromosomes at the bottom of the cell act like a homologous pair even though they are not the same size. These are the X and Y chromosomes that determine biological gender. In beboppers and in humans, the Y chromosome is quite a bit smaller than the X chromosome. How many chromosomes are shown here? I hope you said 8. Now let's model anaphase 1, separation of the homologous pairs. All right, let's move on. Telophase 1, cytokinesis. Now here's metaphase 2. The centromeres are aligned at the equator. Now let's model anaphase 2. Separation of the sister chromatids. All right, there's anaphase 2. Now let's do telophase and cytokinesis to produce our gametes. All right, how many chromosomes in each daughter cell? And they are still daughter cells even though the dad made them. Daughter cell is just the cell that results from mitosis or meiosis. So how many chromosomes in each daughter cell? I hope you said 4. That's the correct number. These are haploid gametes. Let's take a closer look at these gametes. Are these gametes identical? Okay, we've randomly selected one of our gametes to become the sperm. Okay, it's time for fertilization. We have our haploid egg. Let's wait for our haploid sperm. Okay, fertilization occurs when the sperm delivers its genetic material to the egg. Now we have a fertilized egg called a zygote. Through the magic of fertilization, we have a zygote. Remember, a zygote is a fertilized egg. And half of the zygote's chromosomes come from mom, and half come from dad. So notice I've paired up the chromosomes in homologous pairs again. They're not sister chromatids, they're not identical, they're not attached. These are homologous pairs. One chromosome in each pair comes from the mom, and one comes from the dad. How many chromosomes in our zygote? 1, 2, 3, 4, 5, 6, 7, 8. So we're back to 8 chromosomes. That's our diploid number. And remember, our zygote is going to be diploid just like the mom is diploid and the dad is diploid. So on page 86 of your lab manual, please write down the genotype of the new baby bebopper, shown here, and then determine the phenotype by looking back to the information on page 83. Now, we're going to build the baby bebopper. So make sure you finish the phenotype before you continue on with the video, because I don't want to ruin the surprise for you. You should be able to predict the baby's phenotype exactly based on the genotype. So finish the phenotype, pause the video, and then come back. Okay, so remember, here's our genotype for our baby bebopper. What is this baby's phenotype going to be? Let's take a look. Okay, there she is. So this is a girl bebopper, and we're representing that by putting her on a piece of pink paper. Let's take a look at her phenotype. She's got 3 beautiful eyes, 1 antenna. She has colorful legs. She has 3 body segments. She has a curly tail. So I hope that is the phenotype that you predicted.